Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. This lecture is about molecular genetics. The first lecture is structure, function, and replication of DNA. The objective learning of this lecture are the following. Review the experiments that establishing DNA as the genetic material. Explain the DNA structure, number of strands, polarity, complementary strands, and their antiparallel nature. Explain the steps for DNA replication, strand separation, name of the protein enzyme, pairing of the complementary nucleotides, and termination. Distinguish between the DNA template strand and the new strand. In what direction is the new strand made? Compare and contrast the leading and lagging newly synthesized DNA strands, which is continuously made and which is discontinuously made. We are resemble and differs. Parents, son, daughter, how they resemble each other. And which is responsible for this similarity and differences? It is genetic material or DNA. What is the nature of the genetic material? Is protein, sugar, RNA, or DNA? In a human, the genetic material, of course, DNA. What are the evidences used to prove the DNA as the genetic material? There are many experiments identify and describe the genetic material. One of these experiments, called transformation of the bacteria, carried by Crick in 1928, in which Crick used Streptococcus pneumoniae due to have two strains. One strain, it is called S strain or smooth, produced smooth colonies. That means the cell had polysaccharide capsules. It is caused bacterial function in mice. Another strain called R or rough strain, which lacked polysaccharide capsules, produced rough colonies. In transformation of the bacteria experiments, Crick injected of the S-type into the mice, that it is lead to death of the mice. When injected of the R-type, non-pathogenic type, into the mice, he shows the mice still alive. Also, when the heated cold S-type injected into the mice, also the mice still alive. If the mixture of the live R-type and heated cold S-type cells was injected into the mice developed bacteria identical to that caused by injection of the living S-type. How it is that occurs? The Crick conclude there is something in the S-type bacteria transformed R-type bacteria into the pathogenic type, but he was still unknown which the material responsible for the transformation. The explanation of this transformation, the DNA passed from the S bacteria into the type R, enabling the R to manufacture the smooth coat necessary for infection. يعني هنا تكون المسؤول عن الانفكشن هو الكوتس اللي عبارة عن بوليسكرايد. بوليسكرايد المسؤول عنه هو ال DNA. Crick experiment does not show and demonstrate the DNA is genetic material. Therefore, the U.S. physician Avery, MacLeod, and McCarthy investigated the chemical nature of this transforming factor. They repeated the Griffith experiments by using of the heated cold S type. Then added the non-deadly rough type of the bacteria to the heated cold smooth type. Then added the enzymes that destruction of the carbohydrate, lipids, proteins, RNA, and DNA. They showed in all mixtures the mice are dead only when the DNA was removed from the lysate, the mice still alive. They conclude that the DNA was the transforming factor. What is the purpose or function of DNA? DNA is a play role in stores the genetic information that instructs the cell on which proteins to make. So the DNA makes proteins. Both are biomolecules responsible for the determining all the organism's traits, such as eye color, body structure, and enzyme production. Proteins are responsible for the most of these traits. The proteins it is also codes by the DNA or gene. DNA is a large macromolecules or polymers with three types of component. Sugar is a 5-carbon sugar, pentose in DNA or called deoxyribose, phosphate group, and base. Nucleoside it is composed of base and sugar only. Base is a nitrogen-containing base. It is maybe pyrimidine, cytosine, and thymine in DNA, or uracil and cytosine in RNA, and purine, adenine, and guanine. One base, one sugar, and one phosphate combine to form a nucleotide. The nucleotides combine to form polynucleotide. Watson and Crick proposed the structure of the DNA called Watson and Crick models of the DNA. The major features of that model are the following. DNA is a double-strand molecule consists of two polynucleotide chains running in the opposite directions. Each polynucleotide made up of base, phosphate, and sugar. Those are arranged into the ladder-like structure, twisted to form double helix. The bases are on the inside of the molecule, and the two chains are joined together by double H bond between A and T and triple H bond between C and G. The total amount of the purines, adenine and guanine, equals to the total amount of the pyrimidine. The base pairing is very specific, which make the two strands complementary to each other, and the individual nucleotides in each strand are linked covalently between the phosphate group of the nucleotide and the deoxyribose sugar of the next nucleotide. The sugar and phosphate group form of the outer backbones of the double-strand DNA, and the bases are oriented inward, forming the core of the molecule. So each strand contains all the required information for the synthesis That means replication of the new copy to its complementary. This animation shows that the ladder-like structure twisted to form double helix of DNA. This image shows the structure of the DNA double strand. There are three types of alternative forms of the DNA. The first type, B form, helix. It is the most common form of the DNA in the cells. Right-handed helix contain two grooves. Major groove, it is wide and provide easy access to the bases. And the other groove, minor groove, narrow and provide poor access to the bases. The other type, A-form DNA. It is less common form of DNA. Also right-handed helix. Contain also different groove. Major groove, very deep and narrow. And the minor groove, very shallow and wide. It is a binding for the RNA. The last type is called Z form of DNA. Radical changes of the B form, left-handed helix very extended. It is rich in the guanine and cytosine region. The sugar bases backbone form zigzag. The transformation B into the Z of DNA molecule may play an important role in the gene regulation. The researchers suggested that DNA may replicate in any of the three possible ways: semi-conservative, conservative, and dispersive. In conservative, the one double helix specifying for creation of the second double helix, as a result forming two daughter molecules of DNA, each containing the parental strands and the other containing two new strands. The other types of the replication is dispersive. In dispersive, the double helix cutting into the pieces that would join with a newly synthesized DNA piece to form two molecules of DNA, each of which containing old and new strands. Semi-conservative replication, including three steps. Initiation, replication begins at an origin of replication. Elongation, new strands of the DNA are synthesized by DNA polymerase. And termination, the replication is terminated differently in prokaryotes and eukaryotes. The replication begins at the specific site on the double helix, called origin of replication, in which the two chains are separate from each other when a helicase enzyme breaks the hydrogen bonds between the base pair. DNA is locally opened, resembling a fork that it is called the replication fork. The binding proteins hold the two strands apart, and the other enzymes called primase makes a short stretch of RNA to the DNA template that it is called the RNA primer. The RNA primer was required because the major replication enzyme DNA polymerase can only add bases to the RNA primer at this end. In elongation step, the DNA polymerase attach at the free end of the primer and start to add of the DNA nucleotide. The parental strands act as a template and the new strand grows as hydrogen bonds form between the complementary bases. Beside of its role in elongation of the new strands of DNA, the DNA polymerase play important role in the proofreading activity, checks and replaces incorrect bases. يعني يقوم بالإضافة إلى دوره في إضافة النيو strands أو new bases إلى دور مهم جدا، أيضا بقراءة وتصحيح واستبدال القواعد الغير صحيحة الموجودة على النيو ستراند المتكون. DNA polymerase adding the new nucleotides to the free end of the sugar in the growing strand. That means the replication proceeds in 5-to-3 direction, that it is called continuous strand or leading strand. The replication of the other strand is discontinuous or called lagging strand by forming of the small pieces of DNA. These pieces up to 150 nucleotides called Okazaki fragments on the 5-to-3 template. DNA polymerase begins to remove RNA primer and then ligase seals the sugar phosphate backbone of the pieces building the new strand. This figures shows summary of the DNA replication. بالبداية أول خطوة هو الإنزيم الهيليكيز يرتبط إلى منطقة يسموها بالأوريجين أوف ريبليكيشن. إنزيم الهيليكيز يبدأ عمله لتكسير الأواصر الهيدروجينية ما بين القواعد النيتروجين الموجود على كلا الخيطين، ففي منطقة تسمى ريبليكيشن فورك. الخطوة الأخرى هو البايندينج بروتينز تعمل على فتح الخيطين أو تكون خيطين بعيدة عن بعضها البعض لغرض عمل DNA polymerase. راح يتكون عندنا خيطين، خيط من 3 إلى 5 وخيط من 5 إلى 3. الخيط الأول هو من 3 إلى 5، هذا يسمى بالليدنج. الخيط الآخر يسمى باللاجينج أو discontinuous. يأتي دور البرايميز إنزيم. البرايميز إنزيم يبدأ بإضافة شورت ستراند اللي تسمى بالبرايمر RNA برايمر، تضاف لأن الDNA بوليمريز لا يمكنه التعرف إلى النهاية 3، فقط يتعرف إلى النهاية 3. نرجع إلى الخيط الأول. يأتي دور بعد البرايميز يأتي دور DNA بوليمريز. DNA بوليمريز يبدي بإضافة البيس بيرنج إلى الخيط الأصلي اللي هو يعمل كقالب للخيط الجديد، يبدي بإضافة البيس بيرنج اللي هي مكملات القواعد، مثل الثايمين يضيف لها أدينين، السيتوسين يضيف لها جوانين، وهكذا تستمر الخيط ويتكون الخيط النامي من 5 إلى 3. يسمى هذا الخيط بالكونتينيوس. أما الخيط الآخر ما راح يكون DNA بوليمريز تصنع بشكل مستمر، لذلك يسمى بالديسكونتينيوس. هنا قلنا سابقا بأن إضافة البرايمر أو من قبل البرايميز إنزيم يضيف لنا هذه short strands of RNA at which ال DNA polymerase يتعرف على النهاية 3 ومن ثم يبدي يكمل أو يضيف البيس بيرنج كما في الخيط السابق، لكن بشكل متقطع أو شكل قطع صغيرة. تسمى هذه القطع الصغيرة بالأوكازاكي فراغمنتس. بعد ذلك يأتي دور اللايجيز. قبل لا يأتي دور اللايجيز هنالك الإنزيم البوليمريز يقوم بإزالة البرايمرات الموجودة أو ال RNA برايمرز، ومن ثم يأتي دور اللايجيز يقوم بلصق هذه القطع الصغيرة الأوكازاكي مع بعضها البعض وينتج لنا خيط أيضا آخر من ال DNA. The result of the replication forming of two daughter molecules, each of which containing the old strand and the new strand. Question: which are types of the enzymes involved in replication process? Mention the function or functions of each. There are 5 types of the enzyme involved in the DNA replication. First, helicase enzyme. Unwound parental double helix. Binding proteins act as stabilize separate strands. Primase enzyme adds short primer to the template strand. DNA polymerase bind the nucleotides to form new strands. That means added of the base pairing nucleotides. Finally, ligase enzyme. Ligase play important role in the joins Okazaki fragments and seals other nicks in the sugar phosphate backbone. Thank you very much.